are G protein coupled receptors, or GPCR, with our G protein, which consists of three subunits, are alpha subunit, beta subunit, and gamma subunit. This process starts with the ligand binding to the GPCR, which will prompt a conformational change, which causes the alpha subunit to exchange GDP for GTP, which results in the alpha subunit to dissociate itself with the gamma and beta subunits to go and bind with a membrane-bound enzyme to activate a cellular response. And then, after the cellular response has been sent, the GTP is going to hydrolyze to GDP, which creates a phosphate, and then the whole process will start over again. Our next cell signaling system is receptor tyrosine kinases. So here we have two signal molecules binding to both of these proteins, which are going to come together and form a dimer. After this dimer has been made, the tyrosine are going to grab a phosphate out of an ATP and attach itself to one of the 6 tyrosines. It will repeat this until all of the tyrosines have a phosphate. And then, once all of the phosphates have been attached to a tyrosine, an inactive relay protein is going to come and attach itself to one of the phosphates, creating a conformational change in shape, which is going to activate the relay protein and going to send out a cellular response. Right after this cellular response, a different protein could come in and attach itself to a phosphate, resulting in a change in shape and a different cellular response. This last cell signaling system is called ligand-gated ion channel. This process begins with the signal molecule binding to the ion channel, which is closed right now. But this signal molecule can actually control the opening and closing of the ion channel. Opening this will allow all the ions from the extracellular fluid to come into the intracellular fluid, and vice versa.